Sir Mark Walport is the director of the Wellcome Trust, which is the United Kingdom's largest biomedical research charity. The Wellcome Trust spends more than £600 million, which is nearly 7 billion rand, each year to support more than 4,000 scientists with the best ideas around the world. Substantial funding is also devoted to global health research, both in the UK and in developing countries. Before joining the Trust, Sir Mark was the Professor of Medicine and Head of the Division of Medicine at Imperial College London, where he led a research team that focused on the immunology and genetics of rheumatic diseases. He was appointed a member of the Prime Minister's Council for Science and Technology in 2004 and was awarded a knighthood in 2009 for services to medical research. But he particularly is representing the Wellcome Trust, so let me say a little more about that as by way of introduction. Its major programs include research in Kenya, Malawi, South Africa, Southeast Asia, to help combat tropical diseases, the Africa Centre for Health and Population Studies, which is tackling the most pressing population and reproductive health issues in sub-Saharan Africa, particularly HIV and AIDS, and the African Institutions Initiative, which funds 7 African-led international consortia involving more than 50 scientific institutions from 18 African countries to strengthen research capacity and develop scientific leadership for the region. So it is a great pleasure. As you can hear, there is a huge overlap of interest, and particularly around the areas of capacity building and promoting research in Africa. So it's my great pleasure then to invite Sir Mark to share the podium and to give his address.
So as you'll discover over the next few minutes, it's always slightly dangerous to be giving the talk second on a similar topic, but there is the advantage of hearing Francis speak, that it stimulates some thought. And I think there are 3 points and counterpoints that really struck me that came out of your talk, Francis. And the first one was about the partnership that the Wellcome Trust and NIH has had. And as the slides rolled by, one saw how many of them were not just partnerships between Wellcome and NIH, but in fact global scientific partnerships. And I think that there were 2 things that you said that were important. First, it's the ability now to pursue these major projects all the way through to completion in a way that just wasn't possible before. But the second thing is about the openness of data that has come out of those collaborations. And the genomic community, and now the genetic community, have actually transformed the way biomedical science is done. And I think that one of the big challenges now is for the epidemiological community to do the same. Because actually, it's not really fair to patients who take part in scientific studies if their contribution doesn't maximize the advance in knowledge and the potential benefit to future patients. Disease is a combination of nature and nurture. And so this is a diagram where at the top you have those diseases which are predominantly controlled by the environment. So, I mean, you simply can't get flu unless you're exposed to it in the environment. You actually can't get tuberculosis unless you're exposed to it. But as you've heard, there are also genetic factors in your susceptibility. So some people have genetic variants that make them somewhat more resistant to infection with the HIV virus, for example. There is genetic variation in our susceptibility to disease, infectious disease, and also probably important genetic variation in the severity of disease. So the fact that one young person may die of an influenza infection and the majority shrug it off very easily may well have important genetic underlying factors differentiating it. Cancer, there are strong genetic causes, but of course the biggest environmental cause of cancer by a huge margin is of course cigarette smoking. Still a vast problem in many parts of the world. We've then got a whole group of diseases that are in the middle, and most diseases probably sit somewhere in the middle here. So obesity, type 2 diabetes, attention deficit hyperactivity disorder. And then you've got diseases down at the bottom, which are the classic genetic diseases. And alkaptonuria or ochronosis is the most classical of inherited metabolic diseases, described best by Archibald Garrod in his famous Crinean lectures in 1908. And if you are one of the very unusual people, it's a very rare disease, who has the genetic variants that cause alkaptonuria, then you pretty much 100% will get alkaptonuria, which is a disease in which a pigment is produced that's not metabolized normally, and so the skin has this ochre yellow color, hence it's sometimes called ochronosis, and a very astute physician can diagnose it because a baby's nappies turn black, because urine turns black from these patients if it's exposed to oxygen for any period of time. So, cancer, you were right, Francis. One of the important developments is that it's now possible to sequence cancers to completion. And cancers, of course, are different by and large. There are important inherited variants that predispose to cancer, but cancer arises when mutations occur in normally dividing cells that give those cells a growth advantage. And so they may be in growth factors or growth pathways, and they mean that the cells have the potential to grow in an uncontrolled fashion. And this is work of Mike Stratton at the Sanger Institute, who's been one of the world leaders in this area for a long time. And as an early product of the Human Genome Project, he discovered sequencing a cancer called malignant melanoma, which is quite I mean, it's a rarer skin cancer, but it's an important one. He discovered a mutation in that in a gene called BRAF, and that's a growth factor pathway, and the mutations are in fact activating mutations. And whenever you see an activating mutation in a gene involved in a growth pathway, that's a very good candidate as a cancer gene, but it's also potentially a very good candidate for a therapy. But now it's possible to sequence the totality of cancers, and we start seeing how very complicated the variation is in human cancer. So this, in fact, is a melanoma cell line, and what it shows is that, in fact, there are more than 30,000 substitutions in the genome of that cancer cell line. 1,000 insertions, 270 of them in regions that code proteins, 171 which actually change amino acid sequences. Very, very complicated things, cancers, lots of mutations. What can you use that information for? Well, you can start, for example, comparing mutations in different cancers. So melanoma is a cancer where ultraviolet light is very important. It's one of the reasons that you have to be very careful about sunbathing, particularly if you have pale skin. It's one of the reasons that Australia, for example, has run successful campaigns to get people to cover up. And the mutagen there is ultraviolet light. And of course, in lung cancer, the mutagen is related to tobacco smoke. And you start seeing that the patterns of mutations, and you can just see the colors on this pie chart are completely different. So you start seeing different patterns of mutation in different types of cancers. Here's a recent paper from the Sanger Institute looking at a completely different sort of cancer-forming event which hadn't previously been understood, a rather rare cancer in the spinal cord. But here some sort of catastrophic event has happened. And here are the different chromosomes, and you can sort of see that there are all these rearrangements between chromosomes 3, 4, 7, 8, and 9. And in fact, on this, which is a map of the different chromosomes shown in different colors, you can see that the chromosome 5 here has got these short chromosomes. And so some sort of catastrophic genetic event has occurred, probably as a single event in these tumors. And normally an event like that happening in one of our cells would kill it. But this patient was unlucky because the cell survived and had a growth advantage. Now, why does this all matter? Well, it matters because this is now starting to be turned into therapy. And this is the example of a therapy that's being developed by Roche and Plexicon for that disease, malignant melanoma. And here we see two images of a patient before and 15 days after that patient's been given treatment with a drug designed to inhibit activated BRAF. And what you see on the left, and this is something called a positron emission tomographic scan, and it measures the metabolism in tissues, and what you can see is these very active metabolic blobs everywhere, and these are the metastases of a malignant melanoma. And that's a very nasty situation for that patient. And here, 15 days later, you see this extraordinary response with the metastases having melted away. Now, one always has to be extremely careful when you describe this sort of thing for the first time, because one mustn't raise excessive hope. It's a question of balancing the hype and the hope, and I think that's been one of the challenges for the genome science as a whole. This is an extraordinary response, but it isn't a miracle. Patients who are receiving this therapy are starting to relapse because cancers, by their very nature, undergo further mutations, and those that have a growth advantage grow out. So after a few months, many patients are developing further metastases again. And there are some side effects. But nevertheless, this looks as though it's going to develop into an important new, rationally designed therapy for this previously virtually untreatable cancer. So I'm going to finish briefly with infection. I really didn't think that we'd both show this one, but it's good because I'm probably taking slightly too long. So this is the ApoL1 example. And this is, I don't subscribe to supernatural explanations for this. It's just a very good example of host susceptibility, where on the one hand you have protection against an infectious disease, but on the other you have potentially unhappy consequences in terms of susceptibility to kidney disease. I just want to finish by saying something now about the genetics of microbes, because we've talked about the genetics of humans, but the genetics of microbial infections is equally fascinating and equally powerful. So Staphylococcus aureus, 40% of people in this audience carry Staph aureus. And for most of the time, it does none of us any harm at all. But it's quite a versatile organism, and it can cause serious mischief. So it can cause wound infections, food poisoning, toxic shock syndrome, which can be fatal. It can infect the valves of the heart. It can cause pneumonia. And sadly, it's one of the bacteria that quite early became resistant to penicillins. Now, there is a whole new science of infectious disease that is emerging from the study of the genomes of bacteria and viruses. And so, for example, we can look at transmission pathways of bacteria in ways that we never could before. So you can start mapping Staphylococcus aureus and its variation around the world. You can start doing it at a country level. You can start doing it at a hospital level. And this is a map of genetic variation in Staphylococcus aureus. And we don't need to go through all the details, but each of these rows is a different strain of Staphylococcus. And you can see that essentially each of them have a unique fingerprint. Now, you can now start, and this is from a hospital in Thailand, tracking hospital-acquired infections in ways that were never possible before. So here's now a map of the hospital from the sky, and it's just worth saying that we've talked about the sort of miracle of genome science, but there are other miracles going on as well in a way, and one of them is our ability to geographically map things, GPS systems, in ways that were never possible before. So Google Earth, Google Maps provide ways of doing epidemiology, of locating diseases in a way that we could never do before. And so here we have an outbreak of penicillin-resistant Staphylococcus aureus in a hospital in Bangkok. And this could be a hospital anywhere in the world, I should say. And you would think that, using the old techniques, this was all the same infection. But when you actually start doing the sequencing, then you can see, in fact, that there are this ward here, where, in fact, the source of the infection is from the outside world. Whereas the blue spots, the infection is being transmitted from within the hospital. So we can start analyzing infectious disease in hospital in ways we just couldn't have dreamt of doing before. So that's the science. I just want to finish on some of the other sides of this. So there are other aspects of DNA. There's DNA as recreation, and I actually don't mean this to insult Jim Watson in any way at all. But at the moment, knowing one's personal sequence, I think, is something that many scientists would like to do, but it is of more recreational value than any meaning in terms of our own health at the moment. And that's one of the issues for genetics. And as a doctor, I was always taught you should only really do a test on someone for clinical purposes if you can do something as a result of that. And as Francis said, we're still in early days on that. It's very likely that there will be important information, but there's not yet a reason to all go out and have our full sequence done for medical reasons. It may come. DNA is also important in identification. And this is, I think, a very powerful illustration. And this is an American project called the Innocence Project. And this is a project that uses DNA evidence to, in fact, get people out of jail who've been wrongfully convicted. And it's a very good website, and this is the data as of March the 1st this year. You can see 266 exonerated, 947 years in prison, innocent. So DNA is a very powerful tool for forensic purposes. And it can convict people, but it can also free them and prove that they're innocent. We talk about predictive DNA. There are some remarkable things. This is a study in the UK. Men, the surname travels with the male line. So my surname is Walport, my son's surname is Walport. Y chromosome travels in the same direction. So it turns out that actually Y chromosome genetics is quite powerful at predicting your surname. And if you have an uncommon surname, there's a reasonable probability that a sample of DNA will be able to guess the surname. And in fact, there's a well-known example of a child whose father was an anonymous sperm donor. And he managed to track down his father using this technique. Now, you can just imagine the phone call. So there are all sorts of ethical issues. And I think that it's very important that these are discussed and debated. This is fascinating stuff. The newspapers, politicians, the public at large are very interested in this topic. How do we regulate it? It's an international activity. It's going to be very difficult to stop people offering DNA sequencing tests on the web because it is such a global activity. And the pace of scientific advance is extraordinary. So, looking to the future, it's important that we take the public with us, and these are just some examples of all of the headlines that I talked about. And you can see from the number of these headlines, this is very interesting stuff. The media does like it. So what will be the future? Well, I don't think any of us know. It's always much easier to predict the past. Will everyone have their own genome sequenced? Who knows? Will we be able to deduce functional consequences? Almost certainly. Will they be important? Almost certainly some of them will. Will individuals be comfortable about what their sequence means? Well, I think not at the moment, and I think that there's a big job to educate the doctors as well as their patients, because this is a field that's advancing so fast. And will we start to be able to make well-informed choices of medical treatments, and I think in particular side effects? I think in many cases the answer may be yes. So I just want to finish with this slide, because the image on the left is of Sir John Sulston, who led the UK component of the Human Genome Project. He's a very distinguished chap, and as such, the National Portrait Gallery asked if they could have a portrait of him. And so John Sulston decided that this would be his portrait. And this is the portrait of John Sulston that hangs in the National Gallery. And in fact, you can, in the National Portrait Gallery. And in fact, you can just about see, maybe you can't from the back, that there are lots of spots. So in fact, this is clones of John Sulston's DNA. And actually, it's a rather labor-intensive portrait, this, because periodically, the Sanger Institute has to provide new bacterial clones to actually put on this. But my much deeper point is that we are actually much more than our DNA. And my personal preference is for the portrait of John Sulston on the left, which captures the environmental aspects of John Sulston as well as the genetic. And I do think that we must recognize that we are a very complicated mixture of our genes and our environment, and thank goodness for that. Thank you for your attention.